very excited to be here with you guys today.
Thanks.
I'm going to try to share my screen now, and Neha, if you'll just let me know that everything is working.
Yes, ma'am. So far, we see your name in there, so you're good.
Okay, super. So I think you're seeing my screen.
Yes, we do.
Okay, wonderful. So I'm going to show an example today of a plant breeding analysis in barley using JMP Genomics. And I'll talk about a few kind of detailed things, and hopefully we won't get lost in the weeds too much. I just want to show you guys some really cool things that you can do with JMP Genomics. So first, I'll introduce the data. Some of the things we'll do right at the beginning are bring the data into the tool. So JMP Genomics is a hybrid product using JMP, which is a product from SAS, but it is not actually SAS software. So it's a hybrid of this JMP product from SAS and typical SAS software and the genetics capabilities in SAS software. So JMP is going to be used as sort of a visual front end and sort of the hub. And so we'll use it more like pointing and clicking and filling in some questions instead of as a true programming language, but then it's going to call out to SAS and write the SAS programming statements in order to actually do the analysis. So we'll use JMP Genomics to bring some data into that software. We'll do some cleaning. We'll explore properties of some of the markers that we're looking at using for predictive modeling. We'll filter out some bad markers, some bad samples, impute some missing genotypes. Then we'll look at some modeling of those phenotypes, and then we'll do a little bit of progeny simulation. So this is a pretty complicated example I'm going to show you. You can actually use the software for much simpler scenarios as well, like a typical GWAS, if you have genetic data and you want to associate that with some phenotypic traits. You could use much simpler models. I'm going to actually show you kind of the most complex version with some deep predictive modeling options, and then using that to actually do a simulation of what would happen generation to generation if we choose different crosses. So this particular example, we've got 5 plant traits that we're interested in. These are ERG, SSW, TW, PRO, and PY. ERG is a certain chemical, SSW is the seed weight, the TW is the total weight, PRO is the protein content, and PY is the plant yield. So we're going to look at these 5 measurements, and we're going to use the genotypes of the barley to help us figure out which plants to choose to make crosses, and then we're going to actually predict what happens when we do those crosses, and we're going to simulate generation to generation. So by doing this in the computer instead of in the field, we're actually going to be able to do this very rapidly. So the end result is going to be sort of, we're going to get views like this, so we have information here about the mean, the standard deviation, the maximum, the minimum, the range that we expect for each of the traits. So we see that for ERG, for the same thing for SSW, for TW, for protein, and for the plant yield. That was the 5th one. And we can actually make selections. You can see on that side panel, I'm looking at the mean for ERG, the mean for SSW, the mean for total weight, the mean for protein, the mean for plant yield. And I can actually use these sliders, or I can click in there and enter numbers and say, I only want the plants that are going to give me average yields in this range. So as I apply these filters, we can pick out just the points that are black instead of light gray are the points that I'm choosing, and so those correspond to specific plants, and those are the plants I care about making further crosses. So that's kind of the idea. So we're going to look at those distribution of expected traits for all the genotypes. We're going to pick those regions of the traits that are best, and then pick out the plants that give us that. So here's kind of zooming in on that. And then once we do that and we create some simulations, we're going to look at what happens. What do we expect when we make those crosses? So here's an example of crossing a couple of our genotypes. So if we cross the NOS 161 with NOS 62, that's this cross down here, that gives us this purple dot that I'm pointing to. That's our predicted mean ERG level for this plant. And if we cross NOS 62 with NOS 64, that's going to be our predicted mean. So this is making a cross in one generation. We get one predicted mean, and so we can pick out which crosses seem to give us the highest mean, or we could, instead of looking at mean, we could look at the range, or we could look at some other value, some statistic about that. And then as we do this over generations, we can watch what happens when we're looking over here, we're looking for 4 generations in a row. And so we can see what we call a caterpillar plot. You might be able to see why we call it a caterpillar plot. If there is in fact an effect of the genotypes on this trait, if we can actually breed this trait to be better, then we should see from generation to generation as we make these crosses that we see this improvement. So we can see sort of 4 dots that are the same color and they're all increasing as we make these crosses over generations. So how do we do all of this in JMP Genomics? First, bringing in our data, you can start with data in a whole bunch of different formats. Maybe you have an Excel spreadsheet, maybe you have CSV style table of data, maybe you have data coming from other software. There are tons of ways you can bring data into JMP Genomics. So here's one example is sort of bringing in something that's this sort of text style, like an Excel spreadsheet or a CSV. So when we bring this into JMP Genomics, we want to get something that looks like this. We want to get a column that has the ID of the plant genotype. We want something that has a column for each of the markers. So M1 is marker 1, M2 is marker 2. So you might have very different looking names than this, depending on where you're getting these data from, but this is the idea is that these are the markers. So we want this in a numeric format in order to do statistical tests on it. So what we're going to see in those columns of data for the markers, we're going to see 0s, 1s, or 2s. 0s will be that that plant is, I think I'm going to have a note. Nope, I have no note about this. So 0 is homozygous for the major allele, 2 is homozygous for the minor allele, and 1 is heterozygous. So if you've got a different scheme, then we would do the numeric coding differently. But that's what we're going to get when we bring this into JMP. And there's a tool that lets us take whatever kind of version of the data we have and recode it quickly into these numeric genotypes that we need. So once we merge this with some phenotypic data, so we've got the markers in this data set, but we also put in all the information about the phenotypes. Now we're going to have 309 rows. Those are our samples. But 2 of those rows actually don't have the phenotypic information. So you can see those 2 at the top. So we really have 307 complete records. And we want to check the quality of that genotypic data. We might need to do some filtering. So we might want to filter out some of the markers, get rid of markers that don't have good quality information, or get rid of individuals from the data set that don't have good quality information. So there's a tool in JMP Genomics called marker properties. And marker properties lets you explore the properties of the markers and then do some filtering on them. So this is what a dialog box looks like in JMP Genomics. We have a lot of ways to get help. That process description that's hyperlinked at the top. When you're using JMP Genomics, if you click on that, it'll take you to the help about this process. All these little hyperlinked question marks, they take you directly to the help about that particular input. And at the bottom where you see the run, save, load, apply, reset, and cancel, the load button, if you don't know how to fill this out, you can click the load button to find an example and then run an example to see how the example gets filled out. So that load button is actually sort of like an example button in many cases. You can also use it to load things you've done previously. So there's lots of ways to get help here, but what we're doing is we're filling in the name or the location and the name of the data set that we're using. So when we bring it into JMP, we actually save it as a SAS data set, and that extension is .sas7bdat. So you can see this is the folder location and the name of the file that I've saved the data as. When I select this, it populates this box with available variables for all the things that are in that data set. And then I have a number of ways I can specify the markers. One of them is I can just say I want marker 1 up to marker 7,864. And then I tell it an output folder to drop everything in. You can see there were more tabs here. So the general tab is just what's the data set, what are the variables you care about. Annotation, if you've got annotation data to carry through the analysis, you can associate that so that when you do filtering, it will filter the annotation data as well, and everything just stays together through the whole analysis. Options, there's some additional options. Hardy-Weinberg equilibrium p-value plots if you want to request that if you've got an experimental sample. And then some preferences for how your output could be displayed. So when you run this marker properties, you see visuals. That's the point of connecting JMP with SAS, is to get these really nice interactive visuals. So this visual is showing us a plot of the minor allele frequency over all the markers. So some of the markers have a lot of minor allele frequency, and most of the markers have pretty low levels. So we may want to filter on the minor allele frequency. We may want to make a cutoff for the minor allele frequency of 0.05 to make sure that we have at least 5% of the data with the major allele, 5% with the minor allele. The missing genotype proportion plot also shows us we've got lots of data with no missingness, but at about the top 25% of the data has a little bit of missing data, and some of the genotypes actually have almost completely missing data. So we can filter to make sure that we're not missing genotypes for more than 10% of the markers. And then we can ask for, for more than 10% of the samples. We can ask for a cutoff for the Hardy-Weinberg equilibrium p-value, which of course matters for an experimental population, but if you have observed data, this isn't going to be as meaningful. And then we can filter the individuals as well. So if we've got certain individuals that are missing a lot of their marker information, we can filter those guys out as well. So what we've done here is just filter off. We want to make sure we don't have incredibly rare alleles in the data set. That's that first cutoff for minor allele frequency. And then the missing genotypes for markers and for individuals is saying we also want to make sure we get rid of markers that have lots of missing data, and we get rid of individuals that have lots of missing data. So just cleaning up the data, we've got these nice standard workflows for cleaning data. When we do this, the last panel we could look at was that the specific distribution of the individuals that with the missing values. So when we do this, we click on the filtering, and we actually get this really uninteresting looking output. The output is just telling us, okay, I filtered stuff out, and here's your new data set name. So once we've done this, you'll notice that the genotype data that we filtered out just gets an extra little underscore SR added to it from the subset and reordering, because we subsetted it, we filtered it. So all this is doing is pointing us to the new data table. If you choose a workflow in JMP Genomics, you actually don't even have to worry about these new names of data sets. They'll just populate into the next part of the workflow. So it can be even easier than this, where you can ignore these new names and it just moves itself through the analysis. So we see in this little panel up here, when we did that filtering, we didn't remove any individuals. 0 individuals were removed. We did remove 3,455 markers, which left us with 3,542 markers. So we basically took out half of the markers. So half of the markers did not have good enough data for us. So we'll just use the other half of the markers. Then for the markers that are remaining, they might have up to 10% missingness. That was our cutoff. So we might need to impute missingness. So we can use another process called Impute Missing genotypes. Same idea. We fill in we point to where the data set is. It gives us all the variables. We say anything we want to keep in this. So we specifically want to mention the marker variables and then point to a folder. When we do this, we have options about how to impute the missingness, and a typical one is using nearest neighbor samples. So we're basically saying, okay, if there's another line that has very similar alleles for the markers that are nearby, then maybe we impute the same thing that those other similar markers have. So these nearest neighbors are, if there's another line that's very close to this line, then we're probably going to have the same allele for that when we're missing the allele. So we choose our options, we point to where we want that to end up, and so then we have the imputed data. So now we've done all our data cleaning. So it's kind of boring process as far as we're just filtering, we're just cleaning things up, we're just imputing, but we at least get to look at some distributions along the way to get a sense about what's happening. The exciting part now is once we've got data that's cleaned and ready to go, now we can do lots of cool things. So this is the point where we can do a genome-wide association study, so we could look at which markers are associated with which traits. And we can also use more complicated models, so like a predictive modeling review. In that GWAS world, the genome-wide association studies, there's also heritability, so we could look at heritability as well in a mixed model in JMP Genomics. So there's other types of models that we could use here as well. But looking at these predictive models, we again get the same kind of dialog box. We point to the data set, we get a list of our variables, we say, okay, these are the Y variables, these are the responses that I want to predict. So this is the total weight, the seed weight, the plant yield, and the 2 others are in this list as well if we scroll down. And then the predictor continuous variables are those markers. So this way of writing M and then a colon just says everything that started with an M. Those are my predictors. And then I get an option to choose all the different types of predictive models that I might be interested in. So I could choose a distance scoring method, general linear model selection, partial least squares, tree-based methods, quantile regression, radial basis machines, ridge regression, discriminant analysis, k-nearest neighbors, logistic, life regression, proportional hazard. So any of these classes of models. So I've chosen 4 here. And then I would click on each one individually and click edit to make some selections about it. So I'm going to make several different selections within each model class. So what I'm actually going to do is I'm going to end up with 4 models for each of my 4 model classes that I've chosen. I'm going to have 4 ridge regressions, and I'm going to have 4 partial least squares, and 4 partition trees, and 4 general linear models. And what's different between them is I'm going to change internal stuff that they're going to do. So some of the internal stuff, a particularly interesting part of the internal stuff that I'm going to do is the way I've named them, GLM 500, GLM 1000, is that I'm going to do internal choosing in the model to only pick the best 500 traits, or sorry, markers, or the best 1000 markers, or the best 1500 markers, or I'm going to pick a model that uses all. And I'm going to do that same thing for the partial least squares, 500 best markers, 1000 best markers, 1500 best markers, or just use all of them. And there were about 3,500 total. So then we can talk about how to do the cross-validation for this. So again, this is much more complicated than it has to be. This is if we want to do predictive modeling, then we have all of this stuff that we can have at our disposal. But if you're looking for a simpler model, you could also just be doing a heritability study, something with a simple mixed model. And then you wouldn't have this, all these internal things to change and all of this cross-validation to set. This stuff is unique to if you're trying to predict what's going to happen with progeny simulation. So in that case, we really need a predictive model. So we can choose a cross-validation method. Here I'm going to use this k-fold cross-validation and 5 iterations of it. And then the results come out looking like this. It's in a tool we call a jump journal, which it's all hyperlinked things. So this is what you get as a result, and it's just a hyperlink to all the different pieces of results. So you can see at the top, this is for the total weight. So there's 5 pieces of output for total weight. Then there's 5 pieces of output for the seed weight, 5 pieces of output for the plant yield, and so on. The first piece of output for each one is that general linear model, and there's 4 different models in there. The second part is the partial least squares, but there's those 4 different partial least squares models in there. Then the partition trees, then the ridge regression, and then the overall entire cross-validation. So let's look at the cross-validation first. Here's the cross-validation for, which model is this? So this is the total weight. So the total weight model, and here the first 4 things we're looking at are the general linear model. So 500 predictors, 1000, 1500, or all of them. So notice that all of the predictors actually performs worse than using just a subset of the best predictors. So that's interesting. So what we're looking for is high correlation between all, as we did the cross-validation, as we chunked out a holdout set, we want to see high correlation between the model fit and the holdout set. So things we want as high as possible on this axis. The maximum these could be is 1, and you can see our best performer is only about 0.6. So we're not getting perfect prediction, but we're at least getting 60% correlation in our predictions. So that's happening over here for this last model, which is the ridge regression. So the ridge regressions are doing the best. The partial least squares are also doing really well, but the GLM model and the partition trees, they are not performing especially well in comparison. So then we might choose based on this, maybe we're going to use the ridge regression. That was actually our best performer. That's the one on the far right. Our best performer was the ridge regression. Technically, the ridge regression with all of the variables included is the best, but it's only slightly better than any of the other ridge regressions. So maybe we want to save money and time and not use all of the markers and instead just use the 500, which is our small set, or 1000 or 1500. So here in this case, now we look for the equation that predicts, so we're just using the predicted value. So we're using the scoring code from those predictive models, then we're doing this to evaluate the crosses. So as we're evaluating crosses, we're using this tool called cross evaluation, and we have the option of self and non-self crosses, or self crosses only non-self crosses, and some other things to look at. At some point, we can look at simulating the RAL progenies, and right now we're choosing just the non-self crosses. So for the non-self crosses, we again get back to this picture that I showed you way at the beginning, which is a distribution of those statistics about the phenotypes. So the phenotypes, we had that ERG, the protein, the seed weight, and for each of those, we have several statistics. We have the mean, the standard deviation, the max, the min, the range as our defaults of what we show here. So then for each of those statistics, we can apply filters, and we can say, okay, I'd actually really like to move the slider. So if you look at the middle in the data filter panel, we might want to move the slider to select crosses that have high values for the ERG and the seed weight and the plant yield, but maybe we don't care as much about the total weight and the protein content. So if we set the sliders for those higher values of ERG, higher values of SSW, higher values of plant yield, don't really care about TW and Pro, so we don't do anything to filter those, we're going to end up with 15 crosses that we really like. So we're going to use those 15 crosses to produce recombinant inbred lines. Sorry. So here we go. We filtered and chose these 15 dots. So there's 15 dots showing up in each of these plots based on the filters that we put on. So we ignored TW, we ignored Pro, we just put higher values for the ERG, SSW, and PY. So now from those 15 lines, we can ask for the simulating the progenies based on those predictive models. So here I'm going to include self and non-self crosses, and I'm going to use that recombinant inbred line progenies here. And here's what I get as the result of the expected ERG distribution, the expected SSW, expected TW, expected protein, expected plant yield. So these should be a little bit tighter than they were in the whole population. We should be improving slightly, getting a little bit better on ERG, SSW, and plant yield, because those were the things we were trying to maximize. And so here we see that plot we saw at the beginning again, where we can explore which crosses gave us the really high values. So we can see that NOS 161 and NOS 62, that was this really high value, and NOS 62 with 64, that's really good. So NOS 62 was involved in our two best crosses, so that seems really promising. And we can see which other varieties or lines seem really valuable to consider. So with the simulate progenies and that cross evaluation, we can start with doing the evaluating the crosses, pick the best crosses, then do some simulating progenies, then go back, evaluate crosses, do a little more filtering, go back to progeny simulation. So we can do this very hands-on, and a lot of breeders really appreciate being able to do this very hands-on. But there's an even easier way. So instead of going back and forth between all these boxes and filling out all these boxes, we can actually have the computer do this itself. So you just hit a button once, and then it goes through the whole cross evaluation. So if we want to do that, we can, we'll just pick those 15 best we found from our first cross evaluation, and then let it run 4 generations after that. There's one interesting thing that sort of harkens back to SAS code, because we're actually sending this into SAS to run this in SAS. This spot here where it says list of optimization sense for progeny selection, this is asking for what we did before when we said we wanted to maximize 3 of our traits and ignore 2 of our traits. That's what we're saying here. Maximizing is putting 1 in for that trait, minimizing is putting -1, and ignoring it is putting in a 0. So the order of our traits was ERG, SSW, TW, protein, plant yield. So we wanted those first 2 to be 1s, the second 2 to be ignored, and the last 1 to be maximized. So that's what's going on here. That's the one tricky thing about filling this out, is it's sort of like SAS code. So now after 4 generations, we have far fewer points in these plots. So we don't have a cloud of points anymore. And in fact, we have very strong predictions at this point, because we're using our predictive model to figure out what we expect here. So in the end, we can plot those means and watch generation to generation how we're improving on those crosses, how the phenotypes are getting better from generation to generation. So this is a plot of the ERG, and the SSW is below it. So you can see that there's some strong winners in there, that we actually see this strong relationship. For the plant yield, though, we would not see this relationship because the plant yield actually was not very well controlled by the markers. So instead of seeing a nice tight line, we just see kind of random scatter because the plant yield, in fact, was not all that highly correlated with the actual markers. So we just weren't able to predict it very well. So here, the first plot is looking at the correlation for the plant yield, see how low those are. So that's why we didn't get good predictive models. We didn't get good predictions from the predictive models because there just wasn't high correlation for the plant yield. But for the SSW, we did get very good correlations, or again, around 0.6 for the ridge regression model, which is the one we used. So we were able to actually make a difference in our progeny simulation with that phenotype. So again, for the total weight and for the protein, which we ignored, we didn't do any filtering. It just kind of stayed flat over generations. We didn't do anything to try to influence those two. And for plant yield, we also just kind of remained flat. There wasn't a really clear trend, but that was because the markers were not correlated. So I'm going to show you in a second what jump genomics looks like, just to show you very quickly what the tool actually looks like, not just in screenshots, but what we the summary of what we just covered is there's a lot of tools for data import and cleaning, for filtering data to get to the points that actually have not too much missingness, but even when they do have missingness, we can impute the missing values. We did some preliminary analysis, and then we looked at picking a predictive model and actually running with that predictive model and then doing some progeny simulation. Okay, so the last thing that I want to show you, and then I'll take whatever questions, is the actual software. So I'm clicking on this little icon. It has a little helix like DNA and then a little jump man on top of it, a little guy jumping. So that's the Jump Genomics logo. So this is the Jump Genomics starter. And if you've ever used JMP before, a lot of JMP users will use it from the menu bar along the top. So there's options from these tabs along the top, but these same options for genomics are here along the side in this click a category. So typically when you're using JMP Genomics, you start with the import. So if you're importing data, there's a lot of different types of files that you can bring in. So if you had expression data, if this was a different type of analysis, you could find a lot of tools for bringing in data from a lot of different platforms. But very common for genetic data are imputed SNP, either in a tall format or a wide format, and you can use this import tutorial to help you if you're stuck. And then we have actual importers, so things to help you if you've got data coming from Plink or Tassel GBS or something else. So there's a lot of tools to help you bring the data in. Once you've got the data into JMP Genomics, then you might want to move into one of these workflows. So what I showed you was a lot of individual pick and choose things from the genetics, but a lot of those are summarized in one workflow from this workflows option. So I would say when you're starting with JMP Genomics, you start by importing data, then you go to a workflow. So for example, the basic genetics workflow is a good place to start. So it's a little bit of a simpler dialog box. So you point to your data set. Let's see if there's, I'm going to click the load button to see if we can find, here's an example. So it points to the data set. It asks you, what is the prefix for all your marker variables? And in this example, it's G. So let's open the data set and look at that. So here's the data set. It's got some information probably about the phenotypes, and then it has all these markers that start with G. There's 1,000 rows in this data set and 70 columns, and how many of these columns? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 of the columns are phenotypic information, so 60 columns are markers. So we have 60 markers and 1,000 samples. So if we go back to this, so it pointed at that data table, it said G is the prefix of the marker genotype variables. It listed all the others, and here we pick out the trait. Our trait in this case is a binary trait, and it's just, do they have a disease or do they not have a disease? We have two kinds of tests we can do here. We can do a missing genotype by trait analysis, or we can do a case control association test. So the case control association test is what we're looking for here. So disease versus non-disease across all of those markers. And then if we flip through the tabs, if we have annotation, and we do in this case, we can include that here. So we have markers, so we can annotate them by the marker. We know their location. If we wanted to subset before we're looking at the data, we could do that now. If we want to filter before looking at the data, we can do that here. So if we know we want no SNPs with super rare variants, then we can say, okay, I need at least 5% of the minor allele frequency. If I want to set the Hardy-Weinberg equilibrium cutoff, I can do that here. If I want to set a p-value cutoff for actually looking at the plots, I can do that here as well. So when I'm happy, I click run, and it gives you this running process and tells you what's happening. So we're running the basic genetics workflow, and it runs reasonably quickly. Then it shows what it's doing. So it's actually going to do that subset and reordering, that marker properties analysis, another subset and reordering, and then it's going to do the association, the case control association. So I showed you screenshots of the individual components. It's actually even easier to run if you just use the workflows. So that's it. We ran this, and we get the results looking like this. So this is what our results look like, or this workflow journal. If I click on subset and reorder, it just gives us that boring output that says there were 60 original markers. You retained 60. We didn't have to take anybody out. There were 1,000 individuals to begin with, and we had to remove 417 of them. So that was actually very messy. And then we can look at the marker properties. So we can see, here's a Manhattan plot because we have the location, so we can look at them across location. We can look at the distributions of the minor allele frequencies and so on, the missing genotypes, and the individual missing genotypes. So here's those drill down filters again. So if we wanted to change the filters or update something, we could do it here. And then it did the subset and reorder one more time, so it gives us the final data set. And then we go into the analysis. And for the analysis, we see that these are the markers that were significant above our p-value cutoff. So this y-axis has to do, it's the negative log 10 of the p-value. So these are the markers that are important. And if I hover my mouse over them, I see which markers they are. And if I wanted to just pin this so I can keep an eye on these guys, I can sort of customize this graphic. So again, this is why having JMP fused with SAS is so helpful, because you get these really nice interactive displays. You can just grab a corner of this to resize it. You can right click in the middle to find things like changing the color, the markers. So maybe I'll do asterisks instead of dots. So it's really easy to customize things that you're working on in JMP and make these plots look the way you want them to look. I can also use this as a way to rescale it. So that's sort of basically how you're using JMP Genomics. I just showed you a really complicated example in the PowerPoint slides, and then a very simple example with this basic genetics workflow, just to see which markers have differential disease that are differentiating your disease status. So these are the markers that are highly associated with disease. And actually, let's look at a...
Hey, Ruth, do you have room to answer a few questions?
Yeah, now's a great time to take questions.
Okay, one of the first questions that came in was, what other imputation options do you have in JMP apart from the nearest neighbor? I think you can view them in the chat window too, but I want to say it out loud so the rest of the attendees can listen to the question.
Let me see if I can pull up the chat window.
But basically, so the other question is it possible to add pedigree information? And the last question is there a maximum number of markers that can be handled by the software?
Okay, yeah, these are all great questions. Okay, so the imputation options would be under genetics and under, I think it's under genetic marker statistics, is that right? That may not be right. I wonder, let's go to the help and let's see if I can find this on the help. So I just clicked on one of those question marks. Those question marks and hyperlinks will get you into the help, and then once I'm in the help, it's easy for me to just search it here. So if we go to the search and we look for imputation, let's see. don't know why that didn't work. Okay, there we go. So missing value imputation. Impute missing genotypes, that's probably the one we want. So under impute missing genotypes, we have, it will impute numeric missing genotypes 0, 1s, or 2s, so the kind of data I was showing you for diploid organisms using k-nearest neighbors or linkage disequilibrium k-nearest neighbors. So it looks like k-nearest neighbors is the only option for the genetic imputation in this particular context. I think there's more missing value imputation, so let's look at one of these other options because I think that's what's in the genetics workflow, but there may be other. So imputation is performed row-wise. And I don't see anything else on this. So I guess my answer at the moment, it looks like it's only the k-nearest neighbors, those two options, two k-nearest neighbors options. If I can find information on other options, I'll have us post it with this video, and certainly the questioner can send me an email about this directly as well. But it looked from the help like it's only the k-nearest neighbors. The next question was, is it possible to add pedigree information? Yes, it absolutely is. So under the GWAS testing, this is typically, is that where I want to be? Here we go, under other association testing. So GWAS testing is going to include that really simple case control analysis I showed you. It's also going to include slightly more complicated analyses. Here's where we can get imputed SNP trait associations if those are missing. So that's going to be a different type of imputation, just FYI. But GWAS testing is going to include, besides just a simple case control, it'll do a SNP trait association with any kind of trait. It doesn't have to just be disease yes, disease no. So a continuous trait or more categories, any kind of trait analysis here. Under other association testing, here's where you can do SNP-SNP interactions and rare variant things. But the QK mixed models and genomic heritability, this is sort of where I think you're probably interested if you're asking me this question. So the QK mixed model is incredibly useful. I use this for a lot of analyses where I have heritability information. So you can actually estimate the heritability, or if you already have a kinship matrix, so you can either estimate relatedness in a matrix using the data, or if you have the pedigree information, you can include that matrix, upload that matrix in this process, and then model after controlling for that. So the QK analysis is actually controlling for the population level similarity and the familiarity. So if we have a few minutes at the end, I could probably point you to another example about this as well. Sounds good. Is there a maximum number of markers that can be handled by the software? Yeah, that's a good question too. So the JMP aspect of the JMP Genomics software, JMP is an in-memory tool, and then it's using SAS, which you're also using installed on your machine. So when you install JMP Genomics, you're typically installing it in one location, either on a desktop machine, or you might install it on a larger computer somewhere that you're accessing by a server. But whatever it's running on, you're restricted by both the JMP restrictions of data size and the SAS restrictions of data size, which frankly are really great. The JMP restriction is just the RAM of your machine. You basically can't handle a data set that's more than about half of the RAM on your machine. So things that are pretty large are going to run still pretty well. So it's more memory-based restrictions. It's not a number of rows, and same with SAS. There's not a number of rows restriction. It's about the memory and the actual power of your machine to do the calculation. You should, on a good computing environment, you should be able to run reasonably large genomics data sets, but some of these functions are more complicated. So if you just ran a very simple SNP trait association or case control association, that's going to run very quickly. You just saw on a very small data set, it takes a couple seconds. It's not going to be more than minutes or 20 minutes for a really large data set for that analysis. If you run a mixed model analysis, that's going to be much more complicated in the background. What's actually happening mathematically is has many more steps and iterations in it. So that is going to get bogged down very quickly. So we actually have some tools to help speed up some of those slow processes. Like in the QK mixed model, we have an option called K-matrix compression, which is that kinship matrix. So if you're actually estimating that kinship, so here's where we get the Q and the K, we can either estimate them or we can read them in. But this K-matrix compression, it will help it run a lot faster. So there's some tools like that. So sorry that I don't have a clear answer. It's memory based. So you're restricted by the power of your machine, but pretty typical genomics size data sets are commonly run in this software.
The next one is, what are some advantages of JMP Genomics compared to R, and what is the integration like?
Yeah, that's a good question too. So I would say the big benefit of using JMP Genomics instead of R is these workflows that are already built in that make things very simple. Like you just read in the data once, you point to it, you select the options you want, you let it run, and it does all the steps for you. There's more flexibility if you want to use it from the individual components, clicking through different options from below. So then you're creating your own pipelines in that case, and you can actually bundle them into your own workflows if you want to. But the easiest way to use JMP Genomics, and one of the reasons that people often decide to use JMP Genomics, is for the use of these workflows. So these built-in workflows just make things very simple. You just point to the data, you check your options, and it does it all for you. Whereas in R, you're using a lot of programming. So you're going to do a lot more coding yourself, and there's a lot more chances to mistype something or to refer to the wrong data set or to lose which data set you need for the particular thing. If you need to do a lot of switching between tall and wide data, there's just a lot more that you need to do with a software that's not already developed in a pipeline. Another thing about JMP Genomics, besides being a lot simpler to use, is that it's still got a huge amount of really heavy statistical options. So there's still so much power and so many options. There are a few other softwares out there on the market for sort of pipeline analysis, but none of those software packages that I've explored have the ability to do more complicated analyses. Like some of them don't even let you do a three-way interaction in a model. So it's JMP Genomics is both simple-ish to use. I mean, it's still genetic data, so it's not completely simple, but it's this point-and-click sort of based interface and workflows to help you at like pipelines, so that you don't have to keep interacting with things and finding the right data sets and pointing to the right things. It does that for you. But on the other hand, it's pretty deep. The advantage of R is that there may be things in R that aren't in JMP Genomics. Any proprietary software does not have the newest things in it as fast as R does, because someone can write their own R package. So if there's something specific that you want from R, you may want to run some of your analysis in JMP Genomics and then take that clean data back out. So maybe run some of our processing, filtering sort of things, and then take that data back out and do your special thing in R and bring results back into JMP to visualize them. So there's benefits to either, but I think the awesome thing about JMP Genomics is it helps you get started really quickly, get an analysis done really quickly, and then you might go back and make some changes and in how you want to, specifics about your analysis, but you can explore your data really nicely, and you can use the methods much more easily in JMP Genomics.
Another question is, do you have parameter options to modify the tree-based method? On the same lines, the big data, can you support large phenotype sets? And I think you already sort of answered that one. And the last question is, besides the 012 format, can you use an AVH format for markers?
Yeah. So we will change it to the 01 format. So sorry, I'm answering this out of order, but the 01 format is required for JMP to run these analyses actually in SAS in the background. Because we're going to give you a couple of options when you have genetic data, and one of them is a Cochrane-Armitage trend test, and for that we need it to be 0, 1, and 2. We can't just have it be different categories. But you can bring in your data in A, B, in that sort of, like if it's coming from Tassel GBS, no problem, bring it in that way. We're just going to need to change it to the numeric format, so we're going to recode it for you. The question about partition trees and what you can do to set these guys up. So let me take a step back. So again, import and workflows is like the place to start when you're new to jump genomics, but then there's all these components below that you can do advanced things. And I showed you a bunch of these with that barley example. One of those is the predictive modeling options. And I love this predictive modeling review. This is what I used in that example I showed you. If you click on the predictive modeling review, we, under the models section, so again, we fill it out by pointing to the data set and then putting the data variables into different roles. But under the models, this is what I showed you before. So if I take all of these, let's remove all of these models. So I just want to add a partition tree. When I click on it, I can click edit, and then I get all these options within it. So again, I point to the data set, and then predictor reduction. So I can decide, like I did in that example, I wanted ridge regression with the best 500 markers, the best 1,000 markers, the best 1,500 markers, and then all markers. I did that here with predictor reduction. So I could, for example, use a cluster algorithm to do the predictor reduction, or I could use a bootstrap forest or random forest to do that, or I could use a regression technique to do a stepwise regression to get predictor reduction. So there's a lot of internal options. And then for the actual analysis, here's where I can change things like the depth of the tree, the minimum number of observations in a branch, minimum number of observations when I'm looking at categorical values, how am I looking at the probabilities, if my dependent variable is continuous or nominal, I can select the different kinds of trees here. And then even more advanced options that are grayed out here because I haven't selected the right things, but I could make there's so many internal things that I could change. So you can leave it with the defaults if you want to, or you can come here and you can look at all kinds of things for the analysis and for advanced settings within the analysis. Were there more parts to that question?
I think you answered almost all of it. I think there's
Super.
another last one that just came in that does genomics support polyploid marker calls?
That is a great question. I'm so sorry that I don't know the answer to that because I have not looked at any examples with polyploid. I'm a statistician, so my background is completely statistics, and I just really enjoy these applications. So I've gotten involved with some of our developers in looking at some examples and working with some professors on their data. So I'm not an expert in the technical area here at all. I'm an expert in the statistics. So to follow up with that question, I think if the questioner can send me an email, I can get to that answer. Let's see if I can pull up a sticky note or something to type my, where's this? Let's see, let's try a.
Ruth, is it okay to share your email via chat?
Yes, it absolutely is. So I'm going to write it here on the screen. So ruth.hummel@jump.com. That is my email address. And so you're welcome to send me an email if there's something that I didn't fully answer, or you have more questions or questions that didn't get asked here at all. You're welcome to send them to me here.
Thank you very, very much. Well, if you
You're
have
so
no more
welcome.
questions, I think we've almost maxed out our time too. But so glad you could accommodate us. And that was a really nice overview of jump genomics. I'm sure you'll get a lot of questions from the audience.
I look forward to it.
People go on to regret this sometimes, sharing the email. But
no problem.
all of our attendees are plant breeders, and they will very much value your feedback to the rest of the group. So thank you for your time today.
You're very welcome. Thank you for inviting me.
Thanks, everybody.